Hello everyone, welcome to Yucky NEET English Batch. Good afternoon, how's everyone doing? Good afternoon. Yashaswini, Tiso, Tulsi, Ram, Shahid, Meghna, Sneha, Pratibha, Simran, Rimjim, Gire, Savan, Akshara, Navya, Anubhav, Sunandini, Rutu. How you doing? Shubha Madhyana, Vanakkam, Ellargu, Vanakkam. Okay, so yesterday we finished one chapter and I told you that I'll give you the notes for it. I have shared that same PDF of different families on our Telegram group. Hopefully all of you found it. If not, if you were probably in chemistry class at that time, you can download it later. But don't use it as it is. I want you to write it down in your notebooks and make important highlights of wherever there are characteristic features. Alright, I'm doing great. Maliha, thank you for asking. Alright, so today we're going to start a brand new chapter which is called Anatomy of Flowering Plants. Everyone's ready? Also, I got like some other doubt regarding Malvaceae family. I told you in Malvaceae hibiscus family the petals are not fused, and in the floral diagram the petals are not fused, but in the floral formula the petals are fused. The petals, actually that's a mistake. I will correct it in the notes and I will post new notes so you can check that one. Okay? Alright, vanakkam everybody. Cool. So if everyone's ready, give me a thumbs up and we'll get started. Okay? Recap of what, Kirtana? Pavan Sagar is saying that you're doing great. It's obvious that I'm doing great because I'm yapping so much. Alright, cool. So let's get started. Recap of what, Kirtana? We just finished that chapter yesterday. You want me to summarize the entire chapter? That will take me like 40 minutes. Yes, Fabrice, you also have changed in the new notes. You'll get that today. Okay. Cool. Let's get started. Alright. This is a comparatively easier chapter. Okay. I'll disable the comments for a little bit and we'll get started. So today's is going to be the first lecture of the chapter Anatomy of Flowering Plants. This again is exclusively to angiosperms. If you remember when we studied about plant kingdom, we studied in detail about algae, bryophytes, pteridophytes, gymnosperms, and about angiosperms, we finished in less than half a page, right? And then I told you that for angiosperms alone, you have 3 chapters. One chapter we already finished yesterday, which is morphology of flowering plants. And then today we're starting with the second chapter, which is anatomy of flowering plants. You also have sexual reproduction in flowering plants, which we will learn about a little later after your 11th grade portions are over. Okay, so in morphology of flowering plants, we studied about the morphological features of the plant, the features of the plant which you can directly see, external features that can, be studied very easily without making use of any fancy equipment like microscopes. So yeah, that's that. And in anatomy of flowering plants, we're going to study about the internal structure of a plant. We will study about the internal structure of root, stem and leaves. We will study specifically about dicot root, dicot stem, dicot leaf, all of that. So yeah, that's what this chapter is all about. But if you look at the NMC syllabus for NEET 2025, plant tissues is included in the syllabus. Okay. But if you look at the new NCERT textbook, plant tissues is not there. Okay, plant tissues was there in your old textbook. In the rationalized textbook, plant tissues, that part, the tissues, that part has been deleted. So you have to refer to your old textbook for this chapter. Similar to how in morphology you refer to old textbook, even here you have to refer the old textbook. Okay, however, there was one concept in old textbook called secondary growth, beautiful concept, but that has been deleted in your rationalized syllabus. It's also not there in your NEET syllabus. So about that you don't worry. For this chapter, refer the old textbook except for secondary growth part. Whatever is there in secondary growth, that is not there in your NEET syllabus. Everything else apart from that is there. So how are we going to study this chapter? This chapter basically has two parts. In the first part, we will learn about different types of plant tissues. In the second part, we will learn about where exactly those tissues are located, how are they arranged with respect to each other in different plant organs like root, stem and leaves. Today we'll do the first part, which is basically talking about the different types of plant tissues. About plant tissues, it's not anything new. You've been learning probably from 8th grade onwards, or in some state syllabus, I think you have plant tissues in 8th grade. I think in CBSE you have it in 9th grade, I'm not sure. So 1 year, sometime in your high school, you must have studied about plant tissues, all the chyma tissues like Parenchyma, Collenchyma, Sclerenchyma, all of that. So today we will study about that. Alright, so yeah, why am I talking so fast today? I need to calm down. Cool. Let's start off with the definition of tissues. In school, your teachers must have told you that tissue definition is very important for the exam. 1 mark question. Cell definition is very important. And you all might have memorized the definition of tissue already. What is a tissue? Tissue is basically a group of cells. Generally, in a tissue, whatever cells are present, they will have a common origin and they will perform a common function. You've studied animal kingdom in zoology probably, I guess. So over there you will learn about various levels of organization, right? You have acellular level, and then you have loose cell aggregates in porifera, and then you have tissue grade of organization. You have organ level, you have organ system level, different levels of organization. Similarly, in plants you have different levels of organization. Sometimes the plant is not very well differentiated, it is thalloid in nature, but here we are talking about angiosperms. These are very well differentiated plants, and they have specific organs present in them. What are those organs that you see in plants? You see root, stem, leaf, flower, fruit, seed. All of these are considered to be organs of a plant. Okay. So how are these organs formed? Similar to how in us humans, if you consider any organ let's say heart, or our circulatory system, organ system for that matter, heart, stomach, whichever organ you take, you have a epithelial tissue, you have muscular tissue, smooth muscle is present in your stomach, cardiac muscle is present in your heart, and then you have connective tissue that is covering and forming a protective layer on the outside. So you have different kinds of tissues coming together to perform one function. So similarly, even in plants, you have different kinds of tissues that are present in each organ, right? So in the root, you have some types of tissues. In leaves, you have some types of tissues. In the stem, you have different types of tissues. So many different types of tissues make up these organs. Now, what are these tissues in turn made of? The tissues are composed of cells. Tissues are basically a group of cells performing the same job. It's like a group project. Like, if you given a group project in school or college, you would have done it like there is more than one individual involved, right? In a group, there is more than one person involved. That is when you call it a group project. Otherwise, it's an individual project. In a group project, how? To perform one function, to give one presentation, to make one model, multiple individuals are involved. Like that, to perform one function, different cells are involved. Okay, so a group of cells that are doing one particular job, that are performing one particular function is what we call a tissue. Okay, so generally these cells forming a tissue will have a common origin. Origin means how it arises, like from where it arises. That is its origin, and it will have a common function. In some types of tissues, all of the cells in the tissue will be of the same kind. In some tissues, different types of cells are involved. Okay, so we'll learn more about it as we move on. For now, for starters, what are tissues? Tissues are a group of cells, and these cells generally have a common origin and they perform a common function. Okay, and what do these tissues do? What is their role? Each different tissue has its own function to perform. Different tissues come together to form organs in a plant like the root, stem, leaves and all of that. Okay. Now, we'll learn about the different types of tissues. In today's class, we will focus only on the different types of tissues. So, broadly, you can classify tissues into two types, meristematic and permanent. Okay. So, broadly, plant tissues can be classified into two types. The first one being meristematic, and the second one is permanent. Okay. So, what is the criterion for classifying tissues into meristematic and permanent? You look at a tissue, you take a tissue, you look at its cells. Does the cells in that tissue have the ability to divide, or have they lost the ability to divide? In a plant, every cell will not retain the ability to undergo cell division. Similar to how in our body, our heart muscle cells rarely ever divide, our neurons rarely ever divide. Even in plants, specifically in angiosperms, all cells will not have the ability to divide. They would have exited the cell cycle at G1 and gone and rested in G0 phase. Okay, so they're not actively dividing. So some of these cells have the ability to divide. Some of them don't have the ability to divide. That itself is the criterion for classifying these tissues, nothing fancy. Okay, can it divide, can it not divide? That's the only question we're asking here. So what is this classification based on? This classification is based on whether the cells in that tissue have the ability to divide. If they do have the ability to divide, we call it meristematic tissue. They don't have the ability to divide. They had it, but they've lost the ability to divide now. Then we call it permanent tissues. Excuse me. So these meristematic tissues, they have the ability to divide. Since they have the ability to divide, they will keep on dividing. They are constantly dividing. And the cells in this tissue are actively dividing cells. That means they are constantly dividing. One cell gives rise to two cells, two give rise to four, four to eight, eight to 16, 16 to 32, and so on. Okay. So if you remember in the morphology of flowering plants chapter, we spoke about the different regions of a root. In the different regions of a root, we spoke about the root cap, which is a thimble-like structure, protective in function. Right above that, you have a meristematic region. The region of meristematic activity. Over there, the cells are constantly dividing, right? Do you remember the term meristem from that chapter? I hope you do. So, yeah, in the meristematic tissue, cells are constantly dividing. What is the function of this tissue? Why are the cells constantly dividing? In order for growth to take place in a plant, number of cells has to increase. As the number of cells increase, the plant will grow. How will the number of cells in a plant increase? Cells can only arise from pre-existing cells, right? So the cells that are already there, they will start dividing, giving rise to more cells. As a result of that, as a consequence of that, you can see the growth of the plant parts, okay? So meristematic tissue basically helps in growth of the plant. What about permanent tissues? Permanent tissues have lost the ability to divide. They don't divide. Very rarely they might, but in most cases, they will not divide. Again, they have lost their ability to divide. And they have taken up certain permanent functions in the plant. In a plant, there are so many functions that need to be performed, right? Just like how in us we have 4 different types of tissues. The neural tissue will help in coordination. Our muscular tissue will help us in movement and locomotion. Our, what else do we have? We have our epithelial tissue, which is protective in function. We have a connective tissue that keeps our different organs, tissues together. It connects different parts of the body. So like that, we have different tissues performing different jobs. If you take blood, it is involved in transport of nutrients and respiratory gases across different parts of the body. Our excretory system will also have certain tissue that is capable of reabsorption of water and other essential minerals. So similar to how different tissues perform different functions in us, even in plants you have different tissues that perform different functions. I told you that the function of meristematic tissue here is to help the plant grow. Then what is the function of the permanent tissue? You know, some parts of the plant photosynthesis have to take place. There permanent tissues will play a role. Plants they do not have bones like us. So if a plant has to stand upright, it needs certain mechanical support. Some tissues provide mechanical support. If it is an aquatic plant, it needs to have some mechanism which will help that plant float on the surface of water so that it can get sufficient sunlight. So one kind of tissue helps in floating for the plant. And there will be some kind of tissue that helps in conduction, similar to how we have blood vessels that conduct different components like your nutrients and oxygen and carbon dioxide. In plants, conduction of water is important, conduction of food is important. So there are specific tissues that do those jobs. All of these tissues are permanent tissues. They are not dividing, they have stopped dividing, and now they have taken up certain specific permanent functions in a plant. Okay, so that is what we refer to as permanent tissues. You can think of these meristematic tissues as somebody who's in 10th grade or 12th grade right now. Right now in front of them there are many career options, right? So they could become lawyers, they could become doctors, they could become teachers, they could become singers, they could become so many different things, pilots, or they could become like a motivational speaker. I don't know, there are so many options right in front of them. But once you choose your stream, once you decide in your 10th grade that you want to be a doctor, you will take up PCB in college, and then in 11th and 12th, and then you will prep for NEET, and then you will give the NEET exam, and then hopefully you will get into a medical college, and then that is how it goes on. Usually in most cases what happens is, very rarely we see students switching career, right? So after, when there are so many options, they will either choose one and they will become that, they will pursue that particular career. For example, I've spent so much years of my life, like probably 20 years of my life, preparing to become a teacher. Reading and doing research and coming up with things to simplify concepts. I've put in the effort in order to become a teacher, right? Right now, I do not want to become an actor. Like usually if I've chosen this path, very rarely you see that they are switching professions, right? A doctor would have put in so much effort, so much hard work, so many years of his youth into preparation and becoming a doctor. Suddenly one day a doctor will not decide and say, okay, today I feel like becoming a tailor, I want to be a tailor. That rarely ever happens. Of course, of late you're seeing people switching streams very often, but generally, majority of the cases, once you take up a particular function, once you take up a particular career path, you usually pursue it. You do not want to switch, right? Very similarly, here also in our cells, this meristematic tissue can become any type of permanent tissue. Okay, so if you had like 100 meristematic cells present, of them 50 will continue to keep dividing. The remaining 50 will become permanent. Of this 50, some of them will become parenchyma, some of them will become collenchyma, some of them could become sclerenchyma, some will become xylem, some will become phloem. So it will change, it will become one of those permanent tissues. After it has become a permanent tissue, after one of these meristematic cells have matured and become a parenchyma cell, after 2 days it's like, it won't be like I don't like this job anymore, I want to become collenchyma today. That will not happen. Okay, so for the rest of its life it's going to continue as parenchyma. Same thing for others also. In rare cases, in certain plants, you see that parenchyma will become meristematic again, and after that it can become collenchyma, sclerenchyma, xylem, phloem, whatever. About that we learn in another chapter called plant growth and development, like how in society we see of 100 people, probably one person might change the stream later in life, but majority of them usually stick to whatever stream they've chosen. Okay, so meristematic tissue, permanent tissue, primary differences: can it divide, can it not divide? If it can divide, it's meristematic. If it cannot divide, it has lost the ability to divide, it is permanent. What is the permanent function of the permanent tissue? You have so many types there. Depending upon what job it does, you have different types of permanent tissues. Okay, more about it we will learn as we move on. Up until this point, whatever I've spoken, have you understood? Cool. Let's move on. Now let's first talk about meristematic tissue. This meristematic tissue first, and then we'll talk about Permanent tissue. Okay, we'll start off with meristematic tissue. Okay. Yeah. I told you that meristematic tissue is not present everywhere in the plant. There are only certain regions in a plant where meristematic tissue is present, where cells are actively dividing. In most part of the plant, cells have taken up permanent functions and become mature. But in some places, meristematic tissue will be present. Okay, so how did we arrive at the term meristematic? What does that mean? Where did that word come from? It comes from a Greek word called meristos, which means divided or dividing. Since these cells are constantly dividing, we call them meristematic tissue. And the regions in which you find meristematic tissue, specialized regions where you find meristematic tissue, those regions are called meristems. Meristems are not stems alone, okay. Meristems are those regions where you can find meristematic tissue. You can find that in the stem, you can find that in the root, you can find it in different parts of the plant. Wherever in the plant you find meristematic tissue, that region, that specialized region is referred to as meristems. Meristems are not restricted to stems, okay. Now, depending upon the location where these meristematic tissues are located, you can categorize them into three different groups. The first one is apical meristem, the second one is intercalary meristem, and the third one is lateral meristem. We'll learn about each of these one by one. The first one is apical. We already know what apex means, right? So what does apex mean? Tips are called apex, right? So this meristem, according to its name, is present in the tips of plants. It is present in the tip of the root and it is present in the tip of the shoot as well. If it is present in the tip of the root, we call it the root apical meristem. If it is present in the tip of the shoot, we call it the shoot apical meristem. This is a diagram that you have in NCERT. If you remember, when we studied about the different regions of the root, we spoke about how there is a thimble-shaped structure present at the tip of the root. We call it the root cap. Right above that, you have the meristematic region where you have the root apical meristem. Okay, coming to the stem. If this is a stem, this is the tip of the stem. Take a very thin cut the like top 1 or 2 millimeters from the tip of the stem. Look at it under the microscope. Here is what it will look like. Okay, so what is it that we need to observe here? This is the actual tip. Though this looks like the tip, that is not the tip. This is the actual tip. I'll tell you what those are. At the tip, you will find actively dividing meristematic cells. We call this the shoot apical meristematic zone. Okay, now pay attention. Let's say this is the shoot apex. If you look at the shoot apex under the microscope, you can see small extensions arising from the shoot apex. I've told you about this when I spoke about flower. I told you how does a shoot apical meristem transform into a flower. What exactly happens there? When I told you that, I said that here there is meristematic tissue at the tip. This is our shoot apical meristem. Okay, now what are these projections or extensions that you're seeing from the side? It looks like a cuppa, it looks like a frog, but it's not a frog. But yeah, all of these extensions that are arising from the side, they are basically your leaf primordia. Primordium is singular, primordia is plural. In the future, these leaf primordia will become actual leaves. Okay. So, as this plant grows, as the stem grows, the stem becomes elongated longer and longer. The distance that you have here between two leaf primordia, this will start increasing. The internodal length will start increasing. Alright, so what happens then is, you have your leaf primordia, you have your shoot apical meristem here. You have the leaf primordia that are going to become the leaves, right? There is increase in the length of the stem. Okay. So you can see leaves will start becoming bigger. These will become the leaves. Okay. In between the leaves, whatever stem is present, stem region, that will start elongating. So when this happens, some of the shoot apical meristem tissue, some of the meristematic cells at the shoot apex, they will be retained back over here. See, it was here, right? Now this is becoming bigger. As it's becoming bigger, there will be elongation. This shoot tip will become long like this. Some of the meristematic cells will be left behind near the leaf primordia, like that. Near the leaf primordia, some of the meristematic cells will be left behind. Now these meristematic cells that are left behind near the primordia eventually will become the axillary bud. We learnt in the last chapter that if this is the stem and this is the leaf, the angle between the leaf and the stem, this angle is called the axil, and at the axil you will have an axillary bud, and this axillary bud can either give rise to a flower or to a new branch, right? How is that possible? How is this capable of growth? Because in the axillary bud you have certain meristematic cells. How did the meristematic cells get there? They were present, they were supposed to be present at the tips, right? How did they get here? They got here when the the shoot's apical meristem, some of the cells in that got left behind near the leaf primordia. As the leaf primordia grow and become leaves, the meristematic cells that were present in its axil, they will modify, they will transform into your axillary bud. Okay? Yeah. So this, what you're looking at here in this diagram, over here, this is the stem, this is the leaf primordia. Okay, see, it's arising as an extension of the shoot apical meristem. Here again, you have another leaf forming. At the axil of this leaf, you have a axillary bud here. How did this axillary bud form? From some of the shoot apical meristematic cells that were left behind as the stem started to grow. Okay, so that is about your apical meristem, root and shoot apical meristem. Intercalary meristem is the second type of meristematic tissue. What does intercalary mean? This is too bright pink. I want a nice light pink. Okay. So this is the second one. This is called intercalary meristem. Inter means what? Inter means in between. Inter-class competition, inter-college competition, inter-stream competition, inter means in between, right? So intercalary meristem is present between something. What is it present between? It is present between permanent tissues. So in some parts of the plant, there will be permanent tissues present. In between the permanent tissues like parenchyma, collenchyma, in between the permanent tissues, some meristematic tissue if you find, that kind of meristematic tissue is called intercalary meristem. Basically, they occur between mature tissues. What do we mean by mature tissues? Mature tissues are those that have taken its permanent function. It has lost the ability to divide. Basically, mature tissues means permanent tissues. Okay? So, if it occurs between permanent tissues, you call it intercalary meristem. So, let's take a look at intercalary meristem. Okay, so this is apical meristem, occupies the root and the shoot tip. During the formation of leaves, some cells will be left behind from the shoot apical meristem. That is what forms the axillary bud. I've already told you that. Yeah, intercalary meristem occurs between mature tissues. You can mostly find them in monocotyledonous plants like grasses. Over here, you know, when herbivory happens, herbivorous organisms feed on these plants. If the plant is partially broken, if the plant has to regenerate and grow back, then this intercalary meristem will help in its growth. Okay, so that is what the function of intercalary meristem is. Any meristem helps in growth, but if it's present in between the mature tissue, if the cow that is feeding on this grass will eat this part, then over here you have meristematic tissue that will continue to help the plant grow. Okay, so that is what intercalary meristems do. Next we have last type of meristematic tissue, which is our this is our lateral meristem. Lateral means what? Lateral means from the side, right? So these meristems are present laterally, meaning they are present in the sides of certain plants. Mostly you find this in dicots only, dicot root and dicot stem. Okay, so lateral meristems. These also occur in the mature regions of roots and shoots. You see this in dicot plants as well as gymnosperms. You don't see this in monocots. Here is the lateral meristem. What you're looking at here is one plant. We've taken a longitudinal section of the plant. This is the stem. This is the stem. We have taken a longitudinal section of the stem. So you can see different types of tissues here. On the sides over here, on the sides, you can find lateral meristem. So if this is a stem, on the sides you can find the lateral meristem. Similar to how if you take a pen with a refill, how the refill inside is cylindrical, the pen is also cylindrical. If you cut the pen longitudinally like this, you can see that central cylinder as two lines on the side, right? So similarly here also you see it as two lines on the side if you're taking a longitudinal section. If only you cut this like this, if you cut it transversely like this and look at it, you will see an outer layer of the stem, which is the epidermis. We'll learn about it in next Monday's class. And inside that, you will find a cylindrical ring of meristematic tissue that is called lateral meristem. Actually, it forms a cylinder of sorts inside the stem and the root in dicots. But if you take a section like this, you can see it on the side. Okay, so that's why it's called lateral meristem. Alright, so it occurs in the mature regions of the root and the shoot of many plants. They are cylindrical. Examples for that, they have given fascicular, vascular cambium, interfascicular cambium, cork cambium. Remember these. Earlier, you would study about these different types of meristems in the concept called secondary growth, but right now it is deleted. So don't worry about this, just remember their names. Fascicular vascular cambium, interfascicular cambium and cork cambium. Basically, cambium means cambium is a type of lateral meristem. You have three types: interfascicular, fascicular vascular is also called intrafascicular. When I teach you about vascular bundles, I'll tell you what these are. Right now, you will not understand. Fascicular vascular cambium is also called intrafascicular cambium. Then we have interfascicular cambium, and then we have cork cambium. Okay, about this I'll explain later. For now, just remember the three names, the three examples. Okay, that's about your meristematic tissue. The types based on location. Now, depending upon from when the meristematic tissue is present in the plant, you have two different categories. You know, meristematic tissue gives rise to permanent tissue, right? If a permanent tissue has to form, it has to form from the meristematic tissue itself. As the plant is growing in size, its needs will increase. You will need more leaves that can perform photosynthesis. You will need more xylem that can pump water to the leaves. You will need more phloem that can transport food to different parts of the plant. So as the plant grows, you will need newer parenchyma, newer xylem elements, newer phloem elements. Whatever was present initially in the small plant, that will not suffice. So whenever newer xylem and phloem will form, that will be called secondary tissues. What is existing from the beginning, that is called primary tissues. Okay, so in these meristematic tissues also you have primary meristem and secondary meristem. Okay, primary meristem is those that meristematic tissue which is present from the beginning. Secondary meristem will form later. How does that work? I'll tell you. When you have a meristematic tissue, that means it has the ability to divide. It will give rise to some permanent tissue, let's say. This process is called differentiation. Sometimes, very rarely, sometimes, the permanent tissue will regain the ability to become meristematic. It will regain the ability to divide and become meristematic again. Okay. So the permanent tissue also becomes meristematic sometimes. This happens only sometimes. When a permanent tissue becomes meristematic again, the process is called de-differentiation. This meristem formed later. It was not there initially. It formed later because some permanent tissues de-differentiated. This has formed later because of de-differentiation. So we call this de-differentiated meristem. And this de-differentiated meristem can further give rise to permanent tissues again. And that process is called re-differentiation. First time becoming mature is differentiation. It became mature. It wanted to do, let's say the student wanted to pursue medicine, but then they realize it's not their cup of tea, it's too much hard work, they're not willing to do that hard work. They go back, explore other options again, and then they think fashion designing is something that requires a lesser effort, so they start studying for fashion designing course. Okay, so something like that. It was meristematic, it took up a permanent function by a process called de-differentiation. Sometimes, not always, very rarely, the permanent tissue will regain the ability to divide and will become meristematic again. That process is called de-differentiation. After getting de-differentiated, it will take up some other permanent function. Over here, this meristem was always there from the beginning. That meristem is called the primary meristem. This de-differentiated meristem formed later. It was not always there. It formed from a permanent tissue. So this kind of meristem is known as your secondary meristem. It will form later. Okay, if it is forming later, you call it secondary meristem. If it had always been there from the beginning of the life of the plant, it is primary meristem. If it's forming later, it is secondary meristem. How will it form later? It will form from permanent tissues by a process called de-differentiation. De-differentiated meristem is your secondary meristem. Okay. So basically your cork cambium is secondary meristem. It forms because of de-differentiation. Interfascicular cambium forms by de-differentiation, that is secondary. Intrafascicular cambium is primary meristem. Okay, more about it as we go on. After I tell you about vascular bundles, I will explain. Okay. So that's about your meristematic tissue. Let me know if you have any doubts. Geometric arithmetic division, Hema, that comes in a different chapter. Okay, it comes in plant growth and development. It can be found in other plants also, Diya. Cool, let's move on then. Now let's talk about permanent tissues. In permanent tissues, again, you have classification into simple permanent and complex permanent. In a tissue, you have a group of cells, right? If all the cells are of the same kind, you call it simple permanent tissue. If you have different kinds of cells, it is called a complex permanent tissue. For example, think of a group project where in one group project, all 6th grade students, like 4 6th grade students, are asked to perform one, prepare one model or something. And then a bunch of 8th grade students are asked to make another project. Over here, in one project, all of them are of 6th grade. All of them are from the same grade. In another project, all of them are from 8th grade, which is basically they are from the same class. All 6th graders in one project, all 8th graders in one project. So, though there is more individuals, all of them are from the same class. On another hand, you can have projects where there are 5 students, 2 of them are from 6th grade, 1 of them is from 7th grade, 1 is from 8th grade, 1 is from 9th grade. Over there, you have students from different classes, but they are doing the same project, right? Similarly, here, the cell type will be different. Different types of cells will be present in that tissue, but they perform the same function. The role that they are playing in the plant is the same, but it is made up of more than one type of cell. Whereas in simple permanent tissues, you have the same type of cell coming into a group and performing the same function. The main criterion for classification here is how many types of cells are present. If in the entire tissue there's only one type of cell, it is simple permanent. If within the tissue you have more than one type of cell, it is complex permanent. Okay, so this, what is this based on? This is based on the number of types of cells. Number of types of cells. Most students get confused. They write sometimes that complex permanent tissues has more cells and simple permanent tissue has only one cell. That is not what it is. It has one type of cell. The number of cell, if there's more than one cell, then only you can call it tissue, right? So here, the same type of cells are involved in tissue formation. Here, different types of cells are involved in tissue formation. So since here all of the cells are similar, we also call it homogeneous type of tissue. Homo means similar. So if this is called homo, what will we call this? We call it heterogeneous. Hetero means different. Okay. Yeah. Yeah. So in permanent tissues, what basically happens is, I told you, permanent tissues arise from meristematic tissues itself. One subset of meristematic tissues lose their ability to divide, they will take up permanent functions and they stop dividing basically. Okay, that's called mature tissues or permanent tissues. Now, if you think about how, depending upon which career path you choose, you will prepare accordingly, right? For example, if you choose to be a doctor, then obviously you have to prepare for NEET exam. After that, you will have to, study about human bodies, human physiology, all of that. And you have to learn a little bit of empathy. You have to learn to be a little bit, understand other people, empathize with them, be kind, all of that. These are certain features that a doctor should have. Then if you choose to become a lawyer, obviously the paths that you take will be very different. You will prepare using other material. If you, prepare, if you decide you want to be a civil servant, you want to give UPSC exams, then that will require a different kind of preparation strategy, right? Depending upon which job or which career path you will choose, you will prepare yourself accordingly. Very similarly, even in permanent tissues, depending upon which tissue it will mature into, it will form certain modifications. For example, if a tissue decides it has to provide mechanical support, in order to provide mechanical support, inside plants there is no skeletal system, they don't have bones and cartilage. So some cell should, if it decides that it should give mechanical support and help the plant stand upright, as a modification, in preparation for that role, the plant cell will literally sacrifice its life. The plant will die. The plant cell will die. Whichever cell decides it has to provide mechanical support, that cell, once it matures, it will lose its protoplast, it will lose all of its cellular contents and it will die. To provide mechanical support, it needs a thick wall. If the plant part is soft, then it cannot provide mechanical support. So the soft part of the cell, the cytoplasm and everything, whatever is present inside the cell, all of that will die. And instead, that cell will have a very thick wall. Because of the presence of thick wall with a very tough substance, that cell will be able to provide mechanical support for the plant. One type of tissue will decide that it has to store food. That tissue will modify accordingly. So it will become spherical and large in size by accumulating more food. One type of tissue will decide it has to perform photosynthesis for the plant. So over there, it will modify accordingly. One will decide that it has to conduct water. If something water has to be conducted through a type of tissue, for example, xylem, there should be an empty cell, right? Because water is being transported against gravity. So what happens in some cells of the xylem, they also will lose their protoplast, they will become empty, line end to end, form a tube-like structure so that water can be transported through it. So depending upon the function, depending upon what it decides to perform, it will modify itself in such a way that it is, capable of doing that job, right? So these permanent tissues, depending upon what it will become, it will modify itself. So all permanent tissues will not have the same structure. In some tissues, you will find the cell wall being very thin. In some tissues, you will find the cell wall being very thick. Some tissues are living, some tissues are dead. So depending upon what its job is, it will modify itself accordingly. Alright, so here in permanent tissues, we will learn about simple permanent tissues first. The chyma tissues are your simple permanent tissues. They are made up of just one type of cell only. You have three types: Parenchyma, Collenchyma, Sclerenchyma. We learn about each of them one by one, starting with Parenchyma. Parenchyma is the most abundant type of plant tissue. You can find Parenchyma in the root, in the stem, in the leaf, in the flower, in the fruit, everywhere you can find Parenchyma. Okay, so it is the most abundant type of tissue that you can find in plants. When we learn about anatomy in our next class, you will be able to understand how Parenchyma is so abundant, because we'll see that Parenchyma is pretty much present everywhere. So this is present in the soft parts of the plant. Of course, like all plant cells, even Parenchyma cells will have a cell wall, but the cell wall is very thin here. Okay, so the cell wall is not very thick, it is very thin, and it forms a major component within organs. Talking about its shape, it comes in so many different shapes. It can be spherical, it can be oval, round, polygonal, elongated. More about this, where what shape is present, in our next class you will understand. So if you can't remember this right now, it's still okay. And generally they are isodiametric. Isodiametric means almost spherical in nature, where the diameter from anywhere it will be the same or similar, nearly equal. That is isodiametric. Like I already mentioned, it does have wall. Like all plant cells, even here the cell wall is made up of cellulose, so the wall is cellulosic and it is thin. Here the cells are arranged very close to each other, they are closely packed. However, some spaces will be present between the parenchyma cells. We call them intercellular spaces. Inter means in between. In between the cell spaces present, that is your intercellular space. Okay, small intercellular space is present. This is present in different parts. If it's present in the leaf, it can help in photosynthesis. If it's present in a root, it can probably help in storage. Somewhere on the stem, it can help in secretion. So, depending upon where it is present, it can have different functions. Okay, so these are some functions of the parenchyma tissue. Next, collenchyma. Collenchyma is a living tissue. Collenchyma can help in photosynthesis if the collenchyma tissue has chloroplasts in it. Not necessary that all collenchyma will have abundant chloroplast. If it has a lot of chloroplast, then of course it will help in photosynthesis. But its major function is to provide mechanical support. In order to provide mechanical support, you need to have thick walls. So this collenchyma will develop a thick wall, but it will not die. These are living mechanical tissues. Okay, so yeah, over here, if you think about the cell wall will thicken at the corners. And see, if this is the young cell, meristematic cell, it has decided to become collenchyma. What it basically does is, initially meristematic cells will have very thin cellulosic cell walls. Apart from the cellulosic cell wall, it will develop a secondary wall thickening on the inside and the wall will become thick. Okay, so this substance that is thickening the wall is generally hemicellulose pectin. Okay, so because of the presence of hemicellulose and pectin, it will become very thick. This can be found as a homogeneous layer or in patches, okay. And shape also is variable here. It can be oval, it can be spherical, it can be polygonal, it will differ. Sometimes they will contain chloroplast. Okay, if they contain chloroplast, it will help in photosynthesis. Its function is to provide mechanical support. Some amount of flexibility also this will provide. It is present in young stem and petiole. So, one important thing about collenchyma, you find it only in dicot plants, only in dicot stems. Okay, only in dicot stems and probably in the petiole of the leaf also you can find. Only in dicots, you don't see this in monocots. Alright, that is about your collenchyma. Here you can see how this is an actual collenchyma tissue, how you see it under the microscope. So here you can see how this is the wall. This is the cell wall of the adjacent cell. This is the cell wall. This is the cell wall. This is the cell wall. Do you see any intercellular spaces here? The intercellular spaces are absent. You don't see them because of the cell wall thickenings. The cell wall material gets deposited in all of these intercellular spaces at the angles, because of which you do not find any intercellular spaces here. Okay? So this is a mechanical tissue, but it is living. It is not dead. It does not sacrifice itself to provide mechanical support. The third type of tissue is sclerenchyma. Very nice kind of tissue. I like this tissue. It is present in the hard parts of the plant. Basically, this is the kind of tissue that will sacrifice itself for the plant. Okay, so this will, lose its protoplast at maturity, develop a very thick wall, so that it can provide mechanical support and help the plant stay upright. So over here in Sclerenchyma, you have two types. One is called fiber. This one is fiber, and this one, spherical one, is called a scleroid. Okay, so these are generally long and narrow. They have thick lignified walls. Lignified walls means in the cell wall, lignin is present. Similar to how I said pectin is present in Collenchyma, lignin will be present in Sclerenchyma. So what basically happens here is, see, initially when the plant cell, Sclerenchyma cell formed, it will be like this. The cell wall will just be this much. Eventually, the cell wall, lignin deposition starts, and this is how much thick the cell wall will become. This entirely from here to here is the cell wall. So cell wall deposition happens on the inside and a thick lignified wall will form. You can see how inside there are no contents, you don't see any cytoplasm or you don't see any organelles, nothing. It is a dead cell at maturity, and whatever space is present inside over here, it's called the lumen. The lumen is almost all obliterated, right? So the lumen is completely covered because of the formation of the cell wall. Okay, so over here in this diagram from NCERT, you can see there are some labels called pits. What are these pits? Pits are regions where the cell wall has not been laid. Okay, so here you can see in white color here, pits are regions where the cell wall has not become thick. Okay, there are certain regions where the cell wall has not become thick. That is called the pit. Over here you can see in these regions the cell wall has not become thick. They are called the pits. Okay, so they have certain pits like that. What is the function of pits? If this cell has to communicate with some other cell, then by means of this pit communication can happen. Okay, so usually Sclerenchyma cells are dead at maturity. That means they don't have protoplast at all. No cytoplasm, no organelles, no metabolic activity is happening here. And its function is to provide mechanical support. And it's of two types, fibers and scleroids. Okay, so this is a fiber, this is the scleroid. Yeah, they occur in groups generally. You can find fibers present in xylem and phloem as well. They have a pointy ends. They are kind of spindle shaped. They have pointy ends. You can find it in different parts of the plant. More about it we'll learn in our next class. Talking about scleroids, where are the scleroids located? That is important for you to know. This also will have thick walls, but this shape will vary. They can be oval, spherical, cylindrical. Different types of scleroids are present. So whenever you eat fruits like guava or pear, you can sense a gritty nature in the pulp of those fruits, right? That gritty nature comes from Sclerenchyma. If you have a microscope, you can just take the pulp of apple or a pear and look at it under the microscope. You will be able to see these scleroids. All right. So it is present in some leaves. It is present in some seed coats. It is present in some fruit walls. It is present in some fruit pulp. It is present in the wall of the fruit of nuts like walnut. Everybody knows walnut, right? It's like the brain. Outside you have a hard fruit wall. In that fruit wall of nuts like walnut, you can find scleroids. In the pulp of fruits like guava, pear, chiku, sapota, in the pulp you can see. In leguminous plants like beans and peas, you can see scleroids in the seed coats. In tea plant, you can see it in the leaf. It's not visible here, but in your notes you'll get it. Okay, so these scleroids, where they are located, remember those locations. Okay, so that's about your scleroids. With this, we have spoken about our simple permanent tissues, three types, parenchyma, collenchyma and sclerenchyma. Let me know if it's clear. Yes, Sahiti, it will be living before maturity. Obviously, it is forming from meristematic tissue. So, meristematic tissue are living tissue. It will die as it matures. So, once the cell wall becomes so thick, see if a cell wall has to thicken, it has to become thick, obviously. To lay new lignin, you need living protoplast, right? Lignin has to be formed, only then it can be sent to the cell wall so that a cell wall is laid. So for laying lignin cell wall, lignified cell wall, cell will be alive all until that point. Once the cell wall becomes so thick and will obliterate the entire space, it will become dead. Intrafascicular is primary, interfascicular is secondary. It'll get disintegrated, Ajay. Red is bright, Srijana. What do you mean? Afroja, what should I explain? There is no question, there is no doubt, no? Intercalary and lateral. Afroja is asking. Intercalary is present in between permanent tissues. Lateral is basically present in the side. Okay. Last part of tissues. It is xylem and phloem. Let's learn about complex permanent tissues. Complex permanent tissues are basically your vascular tissues. Okay. Their function is conduction. Okay. Now, complex permanent tissues is of two types. Both of them are called vascular tissues and they help in conduction. Why these are called complex permanent tissues is because they have more than one type of cell. Xylem has 4 different types of cells. Phloem has 4 different types of cells. Within xylem, you can find xylem vessels, xylem tracheids, xylem fibers, and xylem parenchyma. Of these vessels, tracheids and xylem fibers, these are dead cells. Only xylem parenchyma is living. Okay. If you have to talk about phloem, in phloem you have 4 different types of cells. You have sieve tube elements, you have companion cells, phloem parenchyma, and phloem fibers. Of these, 3 are living, 1 is dead. Fibers are present in phloem as well as in xylem. Parenchyma is present in xylem as well as phloem. Okay. Xylem is involved in conduction of water and minerals. Phloem is involved in conduction of food. Okay, now let's talk about them individually. Okay, it was already listed here, my bad. Yeah, xylem is involved in conduction of water and minerals. Since it has so many dead elements like vessels, tracheids, fibers, all of them are dead with very thick walls. Xylem also provides considerable mechanical support. It has 4 different types of cells which I've already mentioned before. We learn about each of those one by one. Starting with xylem tracheids. Xylem tracheids are these elongated thin cells. They are dead cells. They have thick walls similar to Sclerenchyma. These cells, these tracheids, will line end to end. Okay, how they form a tissue, I'll show you. So if this is one, this will be the other cell, this will be the other cell, like that it will form a long tube. Through this tube, water transport will take place. In angiosperms, majority of water gets transported through vessels. About 5% of conduction of water happens through tracheids. Majority of it happens through vessels only. You can see how in the cells the ends are tapering, which means they are pointed. They have tapering ends. They are dead cells, I already told you. If they are dead, obviously they will not have any protoplasm. Yeah, so inner layers will have thickenings, that is your tracheids. Now, these tracheids are present in gymnosperms as well as angiosperms. Okay, let's talk about vessels. Vessels are characteristic features of angiosperms. Vessels are absent in most gymnosperms. Very few species have it. They act like a connecting link between gymnosperms and angiosperms. So, absent in gymnosperms. In fact, angio means vessel. Seeded plants containing vessels are called angiosperms. You've learnt in body fluids and circulation probably angiogram, angioplasty, all that, right? Angio means blood vessel in zoology. Angio means this vessel here. So similar to how blood vessels are involved in conduction, here these vessels also form tubes similarly and help in conduction. Okay, so angiosperms, angio comes from vessel. So these are found exclusively in angiosperms and not in other groups. Okay, so here you can see they are cylindrical structures. This is a tracheid, this is the vessel. About tracheids I already told you. In angiosperms around 95% of conduction happens through trachea. Vessels are also called trachea. Okay, so if you see, the diameter of tracheids is less, but the diameter of trachea is more. It's wider, the lumen is wider. These are also dead cells only. Inside there is an empty space. So similar cells will lie one on top of another and form a tube-like structure through which water transport takes place. So this also has lignified walls and it has a large central cavity. It is dead at maturity. It lacks protoplasm. So if this is one trachea or if this is one vessel, right, and this on top of it is another vessel like that. Between this cell and this cell there will be this common wall, right? In this common wall you can find pores present, perforations present. Sometimes it will be fully dissolved, sometimes perforations will be present, and through those perforations, water transport will take place. That is about your vessels. Now talking about xylem fibers, we spoke about sclerenchyma fibers. These are the same thing. They have very thick walls. They have a central lumen that is obliterated, that is almost, the wall is so thick that the lumen is almost non-existent, and it may or may not have septa. Okay, fibers, they are arranged one on top of another in within the xylem. Last one is parenchyma. Parenchyma is the only living cell that you find in xylem. If it's parenchyma, obviously it will have a thin wall that is made up of just cellulose, no lignin here. This will help in storage of food, and if any lateral conduction of water has to happen, xylem parenchyma will help in that. What is lateral conduction? When we learn about how these are arranged inside root and stem, I'll teach you. Okay, so this is also involved in storage of food such as starch, fat or tannins. That is about xylem parenchyma. Okay, so here is what it will look like. This is the actual tissue. You have different cells that are coming together here to perform one function of water conduction. Here you have tracheids. Through tracheids, water transport will happen to a minute amount, like a lesser extent. You have fibers. These are the fibers. They are providing mechanical support. The major part here is the vessel. You can see how in the vessel, in the end wall, you have holes present, perforations present. Through that, water transport will happen, and parenchyma is also there. Okay, so that is about your xylem. Next, talking about, oh wait, I have to tell you something. I told you like how you have primary xylem, primary meristem, secondary meristem. Similarly, you have primary xylem and secondary xylem. Primary xylem is present from the beginning, secondary xylem will form later. Okay, in primary xylem, within primary xylem, some xylem elements are formed first, some of them are formed later. Whichever xylem element will form first, over there, the lumen will be smaller. Protoxylem. Okay. So if it is first formed, within primary xylem, if it is first formed, so if you take a section, this is a stem, let's say, or this is actually a root. If this is a root, you take a section of it. Since xylem is like a tube, you will see xylem vessels as circles, right? So if you see here, all the xylem vessels represented by these circles here in red color, they are not of uniform diameter. Some of them are small, some of them are big. Whichever ones are small, they are first formed. That is called protoxylem. Whichever forms later is wider. That is called metaxylem. Okay. So first formed is protoxylem. Later formed is metaxylem elements. Talking about phloem. Phloem's function is to conduct food, generally from leaf to root and other parts, but other opposite also can happen. From the root to the stem also it can conduct. Okay. 4 different types of cells. What are those 4 cells? I've already mentioned. In phloem you have many important points. I'll show you what they are. Just give me 1 minute. I'll drink some water and come. Okay. Yeah. So the part in phloem which is involved in the actual conduction of food is the sieve tube element. It forms an entire tube, okay. But over here the tube is formed from living cells and not a dead cell. That entire tube is called a sieve tube. Each cell in that sieve tube is called a sieve tube element or a sieve tube member. Why are we calling this sieve tubes? Because there is a sieve-like structure present here. If this is one sieve tube element, this is the next neighboring sieve tube element. Here you have cross walls, right? In this you can find pores. We call that a sieve plate. I'm writing sieve spelling wrong from that time. Sieve plate. Okay, so each one is a sieve tube element. Together, the whole entire thing is the sieve tube. Okay, one important point here, sieve tube elements are absent in gymnosperms. Instead of sieve tube elements, they have sieve cells instead. Where what is important, where what is not present, absent, that is something that you should remember. Okay, they are arranged longitudinally as you can see clearly. Now, these sieve tube cells function is to translocate or help in the conduction of food. You know, nucleus occupies a large portion of the cell's protoplasm, right? So these cells, when they mature, to make way for food to be transported through it, to make sure there is less resistance, they will lose their nucleus. Similar to how in red blood cells you see at maturity there is no nucleus, even in these sieve tube cells they are enucleated. They don't have a nucleus. Alright, so at maturity they do not have a nucleus. Where is that point? It lacks a nucleus. So if this lacks a nucleus, how is its activity, metabolic reactions, all of that control? If it doesn't have DNA, what is controlling this? Adjacent to every sieve tube element, you will have its best friend, okay, which will have a nucleus. That is called companion cell. So these companion cells will control the activity of the sieve tube elements. Okay, so yeah, I already told you about sieve plates. They are end walls with pores or perforations. These are associated with companion cells. Its functions are controlled by the companion cell's nucleus. Okay, now talking about companion cells. Companion cells are also absent in gymnosperms. What they have instead is something called albuminous cells. Albuminous cells are present. Okay, so these are parenchymatous cells, but they are slightly specialized. They are closely associated with sieve tube elements. I already told you that. Now, if this is one sieve tube cell, if this one is a sieve tube cell, sieve tube member, sieve tube element, whatever you want to call it, right next to it, you have this companion cell. There is a common wall between the sieve tube cell and companion cell. In this wall, you will find pits. What are pits? I've already told you. Okay, so within this, you will find certain pits, or you can call it pit fields, through which any communication between these two cells can take place. Basically, for transport of food, a pressure gradient is essential. Along the gradient, food will be transported to different parts of the plant. So these sieve tube, in the sieve tube element, to maintain the pressure gradient, the companion cells will help. Okay, you had a chapter called transport in plants earlier. You may have studied in 11th grade if you're a dropper. That in that chapter we used to talk about how exactly water gets transported, how exactly food transport happens via phloem, but right now the chapter is deleted, so you don't have to worry about it. Okay, and then we have phloem parenchyma. Phloem parenchyma is absent in most monocots. Important point again. Okay, it's absent in most monocots. What is parenchyma? They are cells which have thin walls. Over here, the cells could be long and tapering, or they can be cylindrical also. Like all parenchyma, it will have a dense cytoplasm. Like all parenchyma, it will have a prominent nucleus. Like all parenchyma, its cell wall is made up of thin cellulose. Even over here, pits will be present that will help in communication. Remember when we spoke about cell wall in the chapter Cell the Unit of Life, I had mentioned how plasmodesmata will form. Plasmodesmata are cytoplasmic connections. These are different cells that are performing the same job. Imagine if 4 different individuals are performing, working on a project. If they don't talk to each other, will the project be completed successfully? It can't. So if these different types of cells have to function as a unit and make sure that food is transported, they will have to communicate with each other. How do they communicate with each other? By means of chemicals. How do those chemicals reach the other cell for communication? Through plasmodesmata. So basically through the pits, cytoplasm will be continuous between two cells. Through that, exchange of chemicals can happen and communication can happen. Okay, so what is the function of parenchyma? It similar to in xylem. Here also it stores food such as resins, latex and mucilage. Okay, last part, phloem fibers. Phloem fibers are also called bast fibers. Xylem fibers are also called wood fibers. Xylem fiber, where are you? Yeah. So xylem fibers are also known as wood fibers. Actually, these bast fibers or phloem fibers are of economic importance. Okay. So, these are dead cells. If it's a fiber, obviously it's sclerenchymatous. Another important point. Absent in primary phloem, found in secondary phloem. Similar to how you have primary xylem and secondary xylem, you have primary phloem and secondary phloem. This is absent in primary phloem, it's present in secondary phloem. Remember what all we studied in phloem? You started off with the sieve tube elements. That is absent in gymnosperms. Instead of sieve tube cells, you have sieve cells. Then we spoke about companion cells. Companion cells are absent in gymnosperms. Instead of that, you have albuminous cells. Next, we spoke about parenchyma. Phloem parenchyma. Phloem parenchyma is mostly absent in monocots. It's present in dicots. Talking about phloem fibers. Phloem fibers are mostly absent in primary phloem, but it may be present in secondary phloem. These are important points you should remember. Questions, statement-based questions are asked mostly from this. You can take a look at the PYQs in NEET. You'll understand it yourself. Okay. So these are unbranched and they have pointed needle-like apices. We already know that if it's a fiber, that is how its structure is. How is its cell wall? It is thick. At maturity, what happens? It is dead. It loses its protoplast. So this is of commercial importance. The jute, flax and hemp fiber that we get are basically phloem fibers. Okay, similar to how we saw in primary xylem, you have protoxylem and metaxylem. In primary phloem, you have protophlloem and metaphlloem. Protophlloems, sieve tube will be thinner. Metaphlloems will be bigger. Okay, wider lumen will be there. That's it for today. If you have any doubts, please ask. Septate or aseptate. Septate means this is one cell, this is one cell. This wall is common to both these cells. If the wall is intact, it is called septate. If the wall dissolves, there is no cross wall, you call it aseptate. Obliterated means it is taking over the entire space inside. Almost like there is no space left anymore. That is obliterated. Yes, Simran, both of them are absent. Test portions, I think next you will have morphology, not this. Only morphology. What is PAGWS in a day? 5 PAGWS. What does that mean, Kirtana? Companion cell is a good friend because the sieve tube element doesn't have a nucleus of its own. The companion cell's nucleus will help coordinate all the functions in the sieve tube elements. All right. I'll see you next week. Don't waste time. ENDARC, XARC, I'll teach you in the next class, Ramya. When we are doing tissue systems, I'll teach you there. Don't waste time. Study well. In the next class, we will solve some questions. Okay? And you should start preparing for your morphology quiz as well. Thank you, everybody. See you next week. Take care. Enjoy and study hard also. Bye-bye.